Hi, everyone. I'm Neva. I'm a professor at the University of Regensburg and also affiliated group leader at the Helmholtz Institute for RNA-based Infection Research, HIRI in Würzburg, Germany. It's a pleasure to introduce our speakers to you at this RNA collaborative seminar series. So I just have a few comments about the seminar. The question and answers. So each seminar will be about 25 minutes, followed by 5 minutes of Q&A. Regarding the question and answers, I suggest that you type in the answers in the Q&A box and I'll be reading them aloud. Provided we don't have time left, then please also feel free to write comments in the chat window and we'll be able to respond to them. The speaker will also be able to get back to you over email if needed. So with that, I would like to introduce you our two speakers. We have two speakers. First one will be Professor Jörg Vogel, who is the director of the Helmholtz Institute for RNA-based Infection Research since 2017. He's the founding director, and he's also the professor and director of the Institute of Infection Biology, IMIB, Würzburg since 2009. He'll be talking about promise and challenges of programmable RNA antibiotics. And our second talk will be by a postdoctoral researcher at the Helmholtz Institute for RNA-based Infection Research by Daniel Ryan. And he's currently is a postdoc in the group of Alexander Westermann. He joined HIRI in 2018. So with that, would like to get started with our first speaker, Professor Vogel.
Trying to connect. I can't start my video because the host stopped me. All right. Here I am.
Just share again.
Trying.
Okay. Yes, now screen share. Screen share has started. We don't see it yet. Just a moment. There we go. Okay, now we can see it.
Okay. Just need to make sure I can advance my slides. Okay, so hello everyone. It's my pleasure to speak at the RNA Collaborative, in the RNA Collaborative seminar series. It's taken a while to find a slot, but I'm happy to be here. So as I said, I would like to talk about the new direction in the lab, and it's programmable RNA antibiotics. And just by way of introduction, I don't think, I need to convince you that we need antibiotics and we need new antibiotics because antimicrobial resistance is a big problem. And the prediction is pretty bleak. So we're predicted to have about 10 million deaths per year in the year 2050 just from antimicrobial resistance or bacterial resistance to antibiotics. So that's about killing pathogens, but I would also like to argue that we need new types of antibiotics. And that's because of a growing appreciation of the many diverse roles of the microbes that live on and in us, so that colonize our body and that interact with our body and often in a beneficial way. When these interactions between microbes in our body and our body itself go wrong, this might lead to severe disease or it might promote disease progression. And I'm sure you've heard that many of the more than 4,000 different bacterial species that make up our microbiome have been implicated in disease severity of the brain, of the gut, and of other organs. And I cannot tell you whether all these predicted links between an altered composition of a microbiome and a particular disease will stand the test of time. But I think it's been pretty impressive to see how these interactions, predicted interaction, could be recapitulated in mouse experiments. So the big problem here, though, is that most of these species that live in and on our body, most of these microbes are not genetically tractable. So if you wanted to understand their roles, if you wanted to understand how they contribute to health, you would have to have genetic tools to alter gene expression and their activities. Or you would have to have tools to edit the microbiome, editing in a sense that you are able to selectively deplete or modify individuals, species out of these more than 4,000 different species. So how do we normally edit the microbiome? We would do this using phages. This one route, but it's not particularly easy. The other way to take out an individual bacterium is to use an antibiotic. But the problem is, if you wanted to selectively deplete a particular microbe of interest, there's no species-specific antibiotic. So even E. coli, which has been worked upon for many years, there's no particular antibiotic that kills just E. coli without killing other species. So what we have in mind is that we would like to develop programmable antibiotics that can target individual species, and we can design them based on rational rules. And very obviously, being an RNA lab, RNA is our prime target. So what we'd like to do is to target essential genes in target bacteria using short antisense snippets that target mRNAs of essential proteins. And it's an approach that was introduced by Peter Nielsen's lab in Denmark about 25 years ago, even more than that. He's an organic chemist and a co-inventor of peptide nucleic acid. And I need to introduce peptide nucleic acid. It's not a nucleic acid to begin with. It's a chimera of DNA bases that are lined up on a peptide chain, on the glycine chain. But a beautifully base pairs with RNA or DNA, in our case, more important, base pairs with RNA, a very high specificity and a very high melting temperature. So you can use peptide nucleic acid as an antisense molecule and deliver it into bacteria with the use of cell-penetrating peptides that you just fuse to them. And if you target the ribosome binding site, the protein of interest cannot be expressed. And if the protein is essential for the bacterium will die. Basically, this is what you expect an antibiotic to do, to kill the bacterium of interest. So the approach, it's been around for quite some time. There are many reasons why it hasn't been popular, but there are also many reasons why we think it has great promise. It's been shown for several different, to work for several different bacteria. And our main question is in the lab is, can we develop this into a generic platform for antibiotic development? And if you think about the process, you can break it down into multiple stages. You need to be able to deliver your antisense oligonucleotide or oligomer to the site of action, let's say the gut or the eye or any other organ. You need to make sure that your antisense modality, your PNA in this case, you might also use morpholinus, can enter the bacteria. And entering a bacterium isn't that easy because it's got this complex envelope structure with two different membranes, a periplasm in between, but it's also an outer LPS layer. We also need to understand this once you are inside the bacterial cytosol, how do these antisense oligomers find the targets, and how do you actually prevent the production of a protein? What's the best sequence for that? And then last but not least, what actually happens afterwards? So after you've prevented the synthesis of an essential protein. So these are many questions. And as I said, we're primarily interested in developing this into a generic platform to be able to target bacteria that are not genetically tractable. And to begin with, we've broken this down into different questions, asking what's a good carrier, what's a good target, how selective are these antisense PNAs? We know that they work, but how selective are they for the target of for the target of interest? Are they off targets? Is there a bacterial response? What's the best formulation to deliver them And also, are there immune responses? So these are many different questions. And for the time being, we've decided to focus on this. It's basically on getting in and what happens once you have managed to deliver your antisense molecule into the bacterium. So what I'd like to show you is how well these antisense oligomers or antisense antibiotics work. This is data from Salmonella, where a pathogen that's closely related to E. coli, and in this very case, you're using a 10-mer antisense PNA that is shows full complementarity with DNA bases against the the start codon of the ACPP mRNA. ACPP encodes for an essential protein and fatty acid metabolism that's been very popular. It seems to work in many different species. And in our case, we're fusing this PNA to different peptides, KFF, which is basically lysine and phenylalanine, RXR, which is an arginine-rich peptide, or TAT, which is derived from HRV. Okay, so but it's something I want you to keep in mind to interpret RNA-seq to be able to interpret RNA-seq data later. The ACPP gene is co-transcribed with FABF, another gene in fatty acid metabolism. So the expectation is that if you downregulate ACPP, you would also downregulate FABF. ACPP itself, as I told you, is an important protein. It carries intermediates of the fatty acid saturated synthesis pathway or the unsaturated fatty acid biosynthesis pathway through the entire cycle. It's also a very abundant protein and it's conserved to many different species. Okay, so I just want to show you how well this works. And so we're looking at is killing of Salmonella using an ACPP PNA coupled to an RXR, so to an arginine-rich peptide. So if you treat the bacteria and then spot dilution series over time, you can see a beautiful killing of the bacteria starting 15 minutes after treatment. Whereas if you take the same 10-mer sequence and just scramble it, shuffle these bases around, there's no effect on the bacteria. So the PNA or the CPP, the cell point tractor itself, is not toxic. The killing is not only effective, it's also very fast. So if you plot this over time and start with, let's say, 1 million bacteria, then after 15 minutes, there's already a reduction by 2 logs. So basically only 1% of the bacteria survive the first 15 minutes. And then as you continue with the experiment, as you continue the incubation to 60%, you get a million-fold reduction. So the killing is not only very effective, but it's also very fast. Okay, so the interesting question is, it does work, but how does it work? What's actually going on? And surprisingly, nobody's done this experiment in 20, 25 years, neither with microarrays or with proteomics or RNA-seq. So what we decided to do is that to treat bacteria, and then 15 minutes after treatment with either the cell-penetrating peptides alone, the conjugates with the ACPP PNA, the on-target or the scramble control, or the RXR or TAT variants of this. So we'd extract RNA after 50 minutes and then look at what's going on using RNA-seq. And these are, it's a massive number of RNA-seq experiments. Just I want to give you the upshot of this is that what we observe is that, and these are volcano plots telling you the number of genes that are either upregulated or downregulated after a 50-minute treatment. And what we can see is that there's a very selective downregulation of just the ACPP mRNA out of 5,000 different mRNAs. And the FABF RNA, the gene that's co-transcribed with ACPP. So it's very selective and not much else is going on, at least if you use the KFF peptide. So the peptide, the carrier peptide seems to be pretty silent. Whereas if you use other peptides like RXR or TAT, you see a massive induction of genes. And many of the genes belong to envelope stress pathway or to antimicrobial resistant pathway. And what we can deconvolute from this is that these conjugates of PNA coupled to cell-penetrant peptides somehow make it across the LPS layer of these bacteria of Salmonella or E. coli, then just breach the outer membrane, get across the the periplasm, and get sensed at the inner membrane, but miraculously also make it across the inner membrane, and then within minutes or even seconds start targeting mRNAs in bacteria. So that's a simple RNA-seq experiment that for the first time told us what is going on. But the most important take-home message from here is that if you use the right peptide, there's a selective decrease of your target RNA, in this case, the ACPP mRNA. And there's very limited off-target effect on other mRNAs. It seems to be a good tool for selective targeting. Okay. So this is ACPP. Is all of this true beyond the ACPP target? And there's one thing that's particularly strange about ACPP, and at least if you are trained by chemists like myself, ACPP is not only an abundant protein, but it's also a very abundant mRNA. So why would an mRNA that's abundant work so well? So the question we asked is, if you use other mRNAs of lower expression levels, would these be even better targets? So what we decided to do is select different mRNAs of essential genes, going from very high expression that's based on RNA-seq analysis. And in this case, we're using UPEC, uropathogenic E. coli, because we've got saturated high confidence gene essentiality data, so we can really have a good understanding of which genes are essential. We have good RNA-seq data. And which tell us which is a highly expressed gene, and ACPP is one of the most highly expressed genes, even higher than ribosomal proteins, then down to genes which have very low expression levels that have less than 1 copy per cell. So the question we asked is whether there's any correlation between, let's say, transcript abundance and the efficacy of killing, whether it's the efficiency of inhibiting in vitro translation based on in vitro experiments, the predicted TM or mRNA translation efficiency in general, as inferred from ribosome profiling paper data in E. coli. And the sad news is that there's no correlation. Transcript abundance is not a good predictor of a target. It's not important whether a gene is highly expressed or shows low expression level. That doesn't necessarily make for a good target. So we've got many genes with high expression, but again, they afford really good killing. So we didn't really have a good understanding what's a good predictive target, and the same goes for inhibiting in vitro translation. But what we learned in this study, and that's been published in a paper by Linda Propeller, is that there are additional targets, DNAB, FTZ, cell division inhibitor, RPSH, and so forth. They can be used to kill bacteria. We also learned that we can actually go down with the number of bases. We can use 9-mer PNAs, and they are as effective as 10-mers, and in some cases also 8-mers work. And that's important because the shorter, the fewer bases you have, the less likely it will be that you see off-targeting. But then also using RNA-seq for all these different targets, where we've observed that mRNA depletion is not a universal trait. So not necessarily does the block of translation initiation lead to the degradation of the mRNA. The question is what's really important. And in our case, ACPP remains the top target. So where do we go from here? We want to understand what's a good target in order to be able to predict the best PNA for a bacteria of interest. So what we decided to do is to run a really large screen. In this case, these bacteria, E. coli, Salmonella, have got about 4,000 to 5,000 genes, and about 10% of these genes are essential. The numbers vary, but it's usually between 300 to 400 genes per bacterium that are essential. So we looked at the literature and selected targets from global high-throughput screens using transposon insertion analysis from the KO collection and so forth. and decided to pick then about 300 essential genes and designed two PNAs for each of them, usually targeting Shine-Lugano and start codon region, and then carried out a screen in two different bacteria, E. coli K12, which is used in many labs for cloning, but then also in uropathogenic bacteria, which is of clinical relevance. So this is a massive data set, and we've broken it down here. We're not done with the screen entirely, but what I can tell you, what's important to design a good PNA is that you should avoid self-complementarity. That's always a no-brainer, but it's actually surprising that you can see this in 8 mers or 10 mers. Melting temperature does play a role, even though PNA binds very strongly to target mRNAs, but you still have to make sure you've got a high melting temperature, so primarily target GC-rich regions. And then the fitness of the gene and all these other things contributed or are kind of related with this. So self-complementarity, melting temperature, relative fitness of the gene expression levels do play a role, and also the number of predicted off-targets. Okay, I just want to show you something that's really odd. So this is a completely unbiased screen. We just picked 300 targets and then did sub-screening, and they identified a couple of genes that look very promising. These are the ones that show very low minimal inhibitory concentration. So they're down here, for instance. So the longer the bar goes towards the 0.3 micromolar, the better the PNA, the more effective a killer it is. So we started with these 600 PNAs and then identified in the range of 30 promising PNAs with E. coli K12 or 85 in the case of UPEG. And guess what's the best gene? It's really amazing. It's ACPP. So, you know, we've come full circle. ACPP, which was an educated guess in the beginning, also turns out to be the best target from the screen. But it's also good news because it tells us that we have a modern molecule, and what we need to do now is to go back to the drawing board and try to understand why targeting genes in fatty acid biosynthesis is so powerful when you want to kill bacteria. But we also have strain-specific targets, not quite black and white, but good ones that seem to work in one species and not in the other, even though there's no genetic difference between the two, almost no genetic difference. And we've also have several additional targets, additional ones from fatty acid metabolism. So with this, I would like to close and tell you, you know, you can use antisense PNA or morpholinus, in this case PNO, and it would be PNO, for the selective targeting of pathogens. But I think there are many more applications. You could use these to selectively remove root commensals from microbial species. You could alter microbial communities. And this has been done. You could use them to resensitize bacteria to antibiotics by knocking down antibiotics cassette. You can counteract drug inactivation, which is a common problem with the microbiome. So far, we've mainly worked on inactivation or downregulation of mRNAs. You could also think of activating pathways in genetically intractable bacteria. But there's also an interesting application of these antisense PNAs. You could potentially target phages, and there are many more. So it's a very exciting area in the lab, and I would like to thank the people. So the team has grown, and almost, I would say, 70–80% of the lab nowadays work on the use of antisense and the development of antisense antibiotics. It's actually a bigger team based in Würzburg, so it's a collaboration with the lab of Franziska Faber and Lars Barquist, but also Hans Maric and Claudia Hubbard now is organic. Both of them are organic chemists and pharmaceutical biotechnologists. If you're interested in this kind of stuff, we're organizing the first conference ever. We've also coined a term for these antisense antibiotics. We call them azobiotics. It's going to take place in Würzburg in September. It's a small informal workshop. just go on the website Asybiotics 2024 and consider coming to Würzburg. With this, I would like to end. Thank you for your attention. I'm happy to take questions.
Fantastic. Thank you very much, Jörg. We'll start with the questions. So you have about 6 or 7 questions. I think each 1 minute is fair. So anonymous attendee asked if the PNA also binds DNA and how do you target it only to mRNA?
So it's interesting, in the cell it seems to bind only mRNA, and it's an inference from where you get killing. So you really need to sequester the AUG or start codon region in order to see an effect of the antisense PNA. If you go to other regions of the mRNA, to the coding sequence or the 5' UTR, you won't really see any activity. So this is no formal proof that it's mRNA targeting only, but I think that's a very good guess.
Tank is asking, what's the uptake mechanism of PNA by bacteria? Is this mechanism shared in different bacteria?
So it seems that different peptides work better with different species. There isn't a peptide that works for this particular and not another species, but it seems that RXR, for instance, are peptides that get you very well into Pseudomonas. KFF works very well with many species, including cholera and Salmonella. Generally, it's been harder to target gram-positive species, but by now there's a push for alternative carriers, for instance, vitamin B12, which is taken up by bacteria, and more recently siderophores. So these are chelating compounds that are secreted by bacteria to chelate iron and bring it back into the bacteria. And it seems to work very well to use these siderophores and couple them to PNAs or PMOs for active uptake.
Very interesting.
Then yeah, so they cannot actively PNA. These antisense molecules are too large to diffuse into cells. They either need to get across the membrane by some really not understood mechanism, or there has to be active import.
Then Kyung-moon asked how conserved target RNAs are among clinical isolate, not in lab strains.
So, I mean, this is a potential problem. If you, for instance, target ACPP in E. coli, you will also target, you will also kill Salmonella. And this is why we've been running these screens for additional targets, because the more targets you have, the more mismatches you will have. And even though PNA does tolerate 1 or 2 mismatches, the hope is that we'll find targets that differ by 3 or 4 nucleotides, and that will be good enough for selective targeting.
A link to that, Susan Gottesman, hi Susan, asks if you pick up resistant bacteria, mutants in ACPP or other.
Yeah, so there's published literature. 2 groups have done this. One published it for PNA, one for PMO. Both of them miraculously found the same inner membrane transporter. It's an ABC transporter known to import peptide-like antibiotics. It's called SBMA. We've knocked it out in different species. You know, it seems to contribute, but it doesn't seem to be a major factor. We've also done in vitro evolution experiments, looked in targets in 5, 6 different species. And the interesting thing is you rarely ever get mutations in the target site, so in the target gene. Obviously, these, at least with ACPP, the sequence is optimized for ACPP expression. And then other genes, there's really nothing to write home about. There's no, you know, you pick up mutations in metabolic genes that are very hard to interpret, and if you go and make them immune, you won't see resistance. So they seem to be indicative of other changes, which we don't really understand. But the actual reason why we've been doing these screens is because we've been hoping to find transporters and thereby better understand how the PNAs get in, but we haven't.
And last question, if you have time, I'd like to turn to translation expert. Shura Mankin is asking, are targeting initiation region beneficial over targeting the inner codons? I think that's very relevant as well.
Yeah, I mean, for many genes, the inner codons might be even more conserved than the initiation region because of targeting of the proteins. I mean, at least if you look at outer membrane proteins, you know, the first amino acids are usually very highly conserved, and including the nucleotide sequence. But I agree. You know, you just need to be near the start codon, either downstream or upstream. That's the desired target region. Yeah.
Mm-hmm. All right. So there are two more questions about off-target effects and the length of the PNAs, and I recommend that you type the answers in the chat window so that the audience can also see that. Thank you for this excellent talk, Jörg, and for advertising the first ASO conference in Würzburg this September. With that, we'd like to move on with our next speaker, Daniel Ryan. Daniel, hi. Could you please open your video and start sharing your screen? Yeah. Thank you.
Can you see my screen?
Yes, we do.
Okay, perfect.
Thank you.
Okay. Thanks, Neva, and thanks to the RNA Society for organizing this seminar series. So I'm going to talk to you about small RNAs in the model gut symbiont Bacteroides thetaiotaomicron, or B theta in short. Now, this bacteria is a member of the gut microbiota, which is the ensemble of microorganisms that primarily inhabit our large intestine. And the gut microbiota is dominated by two phyla, basically, the gram-positive firmicutes and the gram-negative Bacteroidetes. We are all quite aware that the gut microbiota contributes to our physiology and our development in terms of providing, in terms of metabolism, immune system development, as well as colonization resistance, where it can exclude pathogens from gaining a foothold in the intestine. And at the same time, an imbalance or a dysbiosis of the microbiota it has been implicated in a number of diseases. For example, inflammatory bowel disease, ulcerative colitis, colon cancer, and many others. And as Jörg mentioned earlier, while a majority of the members have been cultured, most of them cannot be genetically manipulated, which is what brings me to our model organism of interest, Bacteroides thetaiotaomicron, or B theta in short. Now, the organism derives its name from the shape of cells as seen under a microscope. So here you have an electron microscopy image. And if you see the shapes of the cells are quite heterogeneous. You have cells that are shaped like the Greek letters theta, for example, iota for the elongated cells, and omicron. So as I mentioned, B theta is a dominant member of the gut bacterial species. It's gram-negative, it's an obligate anaerobe, and it is non-motile. What is also good from a model organism point of view, it is highly tolerant to oxygen, which enables us to work on it a little easier. It has a genome that is 6.26 MB and a 33 kb plasmid that is involved in environmental sensing. However, what makes B theta special is that it is a prolific glycan degrader, and it dedicates about 20% of its genome to machinery known as polysaccharide utilization loci that are involved in degrading complex carbohydrates into smaller fragments, and then which can then be imported into the bacteria and used for energy generation and such. And while Bacteroides, they are inherently resistant to antibiotics, for example, gentamicin and vancomycin, which allows us to select for them, and they are amenable to genetic manipulation. Right. So over the past few decades, bacterial RNA biology and sRNA research in particular, so small RNA research in particular, has essentially focused on pathogenic microorganisms, and rightly so. Pioneering work from a number of labs, for example, has served to shed light on diverse regulators, for example, that play roles and in different stratas, for example, in virulence and in survival to environmental stress, etc. However, there has been little to no emphasis on organisms comprising the gut microbiota. And this is where my work and our work in particular as a group come into focus, where we look at the RNA biology and small RNAs of B theta. Right. And to do this, I primarily employed a technique known as differential RNA sequencing, which allows one to annotate the transcriptome of an organism to a high resolution. So it essentially involves extracting RNA under different conditions of interest. This RNA is then divided into two parts. One is treated with the enzyme terminator exonuclease. So this enzyme degrades processed RNA fragments, leaving primary transcripts intact. And so what happens when you sequence the text-treated versus the text-untreated libraries, you are able to identify transcription start sites, or exactly where transcripts originate within the bacterial genome. And this then allows us to subsequently map, for example, promoter motifs, to look at boundaries of transcripts, to look at organization of operons, and more importantly for us, to look at small regulatory RNAs and non-coding RNAs in B theta. So just to give you a highlight of what we have found, so we identified a total of 4,123 transcription start sites in B theta. And these can be divided into several categories. For example, primary, secondary, internal, antisense, and orphan. And these categories are derived from based on the coverage and location of where they are relative to an annotated coding gene. So for example, primary transcription start sites have the maximum number of reads aligning to them within a 300 nucleotide window upstream of a start codon of a particular gene. And all other transcription start sites within that region would be secondary transcription start sites. And we have antisense transcription start sites, for example, as the name suggests, are those that originate antisense to a coding gene, while you have internal transcription start sites that originate from within coding genes. Right, so looking further ahead, we were able to annotate promoter motifs as well in B theta. We identified the canonical Bacteroides promoter motif that is recognized by the housekeeping sigma factor. And this motif slightly differs from the well-known -10, -30 boxes. We have a -7 and a -33 box in the Bacteroides with a variable spacer region. And we also managed to identify a second motif, which was novel, and we found it to be present upstream of several genes that were involved in the response to oxidative stress. Right, and this also allowed us, so this study also allowed us to resolve the operon structure under a number of conditions, and we were able to show to resolve 522 different operons in B theta. The largest of which belonged to the capsular polysaccharides as well as to the ribosome components. However, now to shift gears to what we are essentially interested in, we were able to annotate 361 non-coding RNAs in B theta. So these are predictions, I must say, and they have we've identified them to belong to various categories. So to all of the 5 categories, namely intergenic small RNAs, cis antisense small RNAs 5' and 3', as well as those that are present within operons. And over the course of the years, we've subsequently able to validate a number of these small RNAs through northern blots, structure probes, and various other biochemical assays. And I would like to talk to you about 2 of them in particular, namely GIB-S and BTMC201 or MAS-B. Just a brief refresher. I know this audience does not need an introduction, but small RNAs are typically, they vary in length from 50 to 250 nucleotides. They often have well-defined secondary structures, for example, 5' stem loops, row-independent terminators. They regulate gene expression primarily by antisense binding to target mRNAs. In some cases, they may also bind proteins, as well as many of the cases, and especially in model organisms that we have looked at, small RNA activity has been shown to be dependent on RNA binding proteins, which facilitate these interactions. And as you will see, and as it has always been shown, small RNAs have often played a role, or they often play roles in bacterial adaptation to environmental stress, to virulence, as well as to responding to the environment. Right. So the first small RNA in B theta that we were able to study and confirm is termed GIB-S. So it's a 145 nucleotide long small RNA with a relatively linear, unstructured 5' region. It has two metastable loops, metastable hairpins, and ends in a 3' row independent terminator. Now, this small RNA GIB-S was found to be highly conserved within the Bacteroides phylum itself only. And we subsequently looked at what conditions, particularly carbon sources, were responsible or were triggers for the expression of this small RNA. And this allowed us to identify a particular carbon source known as GLC-NAC, which is N-acetylglucosamine. And therefore, we term this small RNA as GLC-NAC-induced Bacteroides small RNA. And that's how the name came up. Right. So it's very important to know what targets small RNAs bind to and thereby regulate, because that's how they mediate their function. And in order to try and identify targets, we performed an RNA-seq of a wild type B theta, of a deletion mutant of GIB-S, as well as a GIB-S complementation strain. And looking at differentially expressed genes, we identified 3 genes in particular. So 3 differentially expressed targets in particular, namely BT1871, BT0771, and 3893. Now, further looking into predictions of the interactions between GIB-S and the small RNAs, we were able to validate direct binding between GIB-S and 2 of these targets in particular, namely BT0771 and 3893. GIB-S in these 2 small RNAs binds across the start codon, which is quite typical for small RNAs that normally repress activity, repress translation. And so we were able to ultimately come up with a model for GIB-S, whereby it is a GLC-NAC-induced small RNA that plays a role in repressing or activating various genes involved in carbohydrate metabolism in B theta. Now, Bacteroides or B theta within the gut environment is constantly exposed to various fluctuations. For example, you have various nutrients flowing through, you have interbacterial competition, there is oxygen gradients, as well as the presence of antimicrobial peptides, bile salts, etc. And the next small RNA that I would like to talk about is a result of a study where we tried to look at how the transcriptome of B theta responds to diverse conditions. So we selected a set of stress conditions, for example, acidic pH, aerobic stress, the presence of hydrogen peroxide, for instance, as well as a panel of carbon sources ranging from simple carbon sources such as glucose, arabinose, to more complex sources such as mucin. And we performed both differential RNA sequencing as well as conventional RNA-seq to allow us to further annotate the transcriptome and try and identify small RNAs, for example, that are exclusively condition-dependent that we may have missed, as well as to be able to look at how the transcriptome, both coding and non-coding, responds to these diverse conditions. We also made a lot of this data available, or all of this data, I should say, available to the scientific community via Thetabase. Yes, and so this study allowed us to profile the expression of all of the B theta small RNAs, including a group of antisense small RNAs, antisense RNAs termed PUL-associated antisense RNAs. And these are a very interesting class of RNAs that are or have been implicated to regulate the PULs that I earlier had alluded to, and therefore would comprise an additional layer of regulation to these already highly regulated clusters of genes. Right. Now, in order to try and assign a function to this large set, or this number of small RNAs that we identified, we used our intergenic small RNA annotations of 135 intergenic small RNAs to probe a TN-seq data set that was published from the Deutschbauer group, where they looked at B theta that was exposed to a suite of about 490 or so different conditions, right across the bat, from stress all the way to carbon sources and various other antimicrobial compounds and antibiotics. And what you see here is a result of that study, where on the x-axis we have particular transposon mutants of intergenic small RNAs in particular, and on the y-axis, we have the gene fitness contribution. So a positive value would mean that the particular small RNA mutant contributed to enhanced fitness, while a negative value denotes the opposite. And you will appreciate that while most of these small RNA mutants that we looked at were associated with one or maybe two conditions, one particular or two small RNAs particularly stand out, namely MAS-B, which was found so this mutant was found to have enhanced fitness in a range of antimicrobial compounds, so particularly various antibiotics. And so this was quite interesting to us, and we decided to go ahead and try and see what this small RNA was doing. So I must say MAS-B is a stationary phase-induced small RNA. It is intergenic in nature and is 100 nucleotides in length. We did a co-expression analysis to try and see what conditions or what if there are any known regulators that control or regulate the small RNA, and we found it to be activated by CUR, or the CUR protein, which is a master regulator of carbohydrate utilization in Bacteroides. And it so happens that under starvation conditions, this master regulator is also upregulated. So we went ahead and tried to look at the expression profile of MAS-B in a mutant as well as a complemented strain, and we were able to confirm that, yes, indeed, CUR plays a role in regulating MAS-B. Right. Subsequently, we were also able to validate the phenotype. So we generated an isogenic mutant of MAS-B and looked at its fitness in comparison to the wild type in the presence of either doxycycline and tetracycline. And as you can see, the mutant of MAS-B was fitter and grew better than the wild type under these conditions. Right. So what would be, in order to try and find downstream targets regulated by MAS-B and targets which could perhaps explain this phenotype, we established maps for B theta in particular. And using maps, we were able to identify a target gene for this small RNA MAS-B, namely BT1675. Now this mRNA was identified as having a tetratricopeptide repeat, which has been implicated in assisting protein folding, for example. And we were able to identify and confirm that, yes, indeed, MAS-B does bind and regulate, does bind directly to BT1675, both using MSAs as well as in vivo using translational fusion assays. Of course, with regards to time, I have not shown that data here. So this leaves me with our regulatory model that we currently propose for MAS-B that involves it to be part of the CUR regulon induced under particularly carbon starvation, and it plays a role in repressing BT1675. Now, whether BT1675 is directly a cause for the antibiotic phenotype that we see is still yet to be confirmed, but there is indeed evidence that we are working on. Right. With that, of course, I would like to thank members of the Westermann group, the Bacchus Lab, and Dr. Adam Deutschbauer at Lawrence Berkeley, without whom this study would, of course, not be possible, as well as to HIRI and to you for your attention. I'm ready to take questions. Thank you.
Awesome. Thank you very much for this really interesting talk, Daniel. You're opening up a complete new field in big data, and there are already some questions. One person is raising the hand. I would recommend them to type the answer directly in the Q&A. So the first question is by Quinn Moon. There are more than 100 ECF-like sigma factors, at least in BT5482. Can you link which sigma factors are responsible for the expression of small RNAs?
So we have not yet dived into that, but this is something that could potentially be extremely important, but I wouldn't be able to give you a direct answer of which sigma factors, for example, would trigger the expression. But this is definitely something that we could look into.
Mm-hmm. Another little question from the same attendees. Another just BT5242 strain harbors 4 cryptic conjugative transposons. Did you see any small RNAs from cryptic conjugative transposons under TET-tox-Y inductions? So
in our earlier study, we did find or we did annotate what we believe to be the RTER homologue that is present in the CTN dot conjugative transposon in B theta, but that's as much as I can say. We didn't dive into trying and sort of figuring out what targets it regulates, but we do have evidence that we did find one of these small RNAs that have been previously reported. Yes.
Mm-hmm. I have one question, Daniel, more general. Do you see this as a master regulator of other small RNAs, or do you also see any conservation of the GIP-S? Are these expression levels, or is there any homology to other commensals or pathogenic bacteria?
So both MAS-B and GIP-S, and this tends to be the case for many of the small RNAs that we see in B theta. They are all conserved in the Bacteroides phylum, of course, to different extents in the different genuses. MAS-B is conserved to a less extent in relation to GIP-S, but as to whether one or both are master regulators, I wouldn't be able to tell you because we have not looked at that. But what I can say is MAS-B, as it stands by itself, is a very highly expressed small RNA. And using maps, we've identified 2 targets, one of which I have spoken about over here. But it does not seem to have a very large regulon from what we can see, at least at the moment.
Mm-hmm. All right, thank you. Do we have more questions? I cannot see any, so I'll like to wait another minute to see if any other question will appear in the chat window, in the question Q&A. Again, thank you, Daniel, for this exciting talk. I do not see any further questions coming up, so people are clapping. Thank you, everyone. Evelyn, you raised hand. Could you please type your question, Evelyn, in the Q&A? We cannot see. So I'll just wait until something comes up. So the person is raising still a hand, but I cannot see any question. So please reach out to Daniel over email if you have the question. Evelyn, that's how we could then conclude the session. Thank you, everyone, for attending the HIRI seminar, and we look forward to Singapore seminar, I think the next in the upcoming week. So bye-bye.
Thank you.
Thank you.